Hi, folks. You can thank photosynthesis for every breath you take. The oxygen that we breathe comes from photosynthesis. Today we're going to talk about photosynthesis. We'll start off with a description of the chloroplast, a plastid in which photosynthesis occurs. And then we'll talk about the light reactions and the dark reactions. These are two separate sets of reactions. Photosynthesis is sort of the opposite of respiration. It's respiration in reverse. However, it's not simply the reactions in reverse. Instead, it's two separate sets of reactions, the so-called light reactions and the dark reactions. The dark reactions are not in the dark necessarily. They're simply parts of photosynthesis that can occur without light. They're also called the Calvin cycle. So we'll talk about those. And then after photosynthesis, we'll talk about how plants have adapted to really dry regions. There are plants in dry areas that have a different kind of photosynthesis. And finally, we will turn our attention to how plants respond to environmental stimuli, how plants develop, how plants protect themselves, how plants fend off herbivores, and how they work with light, how they work with gravity, how they work in the world. And they do this through the actions of hormones. So we'll talk about that later. But first, photosynthesis. Let's get going. So, photosynthesis produces the oxygen that we breathe. Photosynthesis is the source of the oxygen in the atmosphere. 50% of it comes from land plants, and about 50% of it comes from marine organisms. These include macroalgae, that is large algae, and they also include single-celled organisms such as microalgae, phytoplankton, bacteria. It is the first step, photosynthesis is, in moving energy into the living world. It is the source of all energy in terrestrial ecosystems. Several kinds of organisms photosynthesize. It's not just land plants. So you can see in the upper left here, land plants, and down from there you can see multicellular algae. These would be macroalgae, probably marine for the most part, and unicellular protists. These are, for example, diatoms or dinoflagellates. The white cliffs of Dover came from these sorts of organisms. Cyanobacteria are bacteria that photosynthesize, and they, of course, were the source of the chloroplast a very long time ago. And finally, there are these organisms called purple sulfur bacteria, and they use sulfur instead of water in the process of photosynthesis. They do not produce oxygen. So lots of organisms photosynthesize. Here is a land plant, and let's zoom in. So the leaf in vascular plants is the main organ of photosynthesis, and that leaf has inside it cells that carry out photosynthesis. You can see here, as we zoom in on a leaf, here's a cross-section. There are chloroplasts right under the upper surface, and they're at the top of the leaf because that's where the sunlight is. And you can see at the bottom there are these stomates or stomata. Either of those plurals is just fine. And they let in carbon dioxide, they let out oxygen. And if we look at one of the cells, one of the parenchymal cells called a mesophyll cell, meso just means in the middle, sort of the middle of the leaf, a mesophyll cell, there it is. And if we look inside, we can find these special plastids called chloroplasts. And here's a chloroplast. They have two membranes, and they have these stacks of thylakoids called grana. And it's in these thylakoids that the first set of reactions occur in photosynthesis. So photosynthesis involves two inputs mainly, carbon dioxide and water. So those are the reactants. And the products are a sugar, which ends up being glucose, so we can say glucose here, water, and oxygen. And in this diagram from your textbook, the authors have labeled the atoms from carbon dioxide in magenta, and they've labeled the atoms from water in sort of a bluish color. So you can follow them here. And one of the first things that should jump out at you is that the carbon in glucose comes from carbon dioxide, the oxygen in glucose comes from carbon dioxide, and the hydrogen in glucose comes from water. The second big thing that really should jump out at you is that the oxygen that we're breathing, the oxygen that sustains the lives of animals on this planet, comes from water in photosynthesis. So the oxygen does not come from carbon dioxide. It comes from water. The process of photosynthesis involves the transfer of electrons. So electron transfer is a redox reaction, that is oxidation and reduction. And so something gets electrons and something loses electrons. If a compound or a molecule is an electron acceptor, it gets reduced. That is, it's reduced in charge because it gains these negative charges. So carbon dioxide in this case is an electron acceptor. Water is an electron donor. It therefore gets oxidized. It loses negative charge, so it gets oxidized. So oxidation is the loss of electrons. And here we go. You can see the same diagram here. So we have one glucose molecule made, 6 water molecules made, and 6 oxygen molecules made for every one glucose. This requires 6 carbon dioxide molecules and 12 water molecules. If we simplify this by subtracting 6 water molecules from each side of the equation, we can get a net reaction that produces 1 molecule of glucose. So here we have energy from sunlight plus 6 carbon dioxides plus 6 waters give 1 molecule of glucose, C6H12O6, plus 6 oxygen molecules. So carbon dioxide becomes reduced and water becomes oxidized. In this process, water is split, it loses electrons, getting oxidized, and the electrons are transferred along with hydrogen ions from the water to the carbon dioxide, and they reduce that to a sugar. So photosynthesis does not occur in one sort of simple set of steps. Photosynthesis instead occurs in 2 separate sets of reactions, and they interact with each other. They supply each other with certain essential ingredients. These are the light reactions, first of all, and the Calvin cycle, or the dark reactions, second of all. The light reactions take place in the chloroplast, and the dark reactions take place in the chloroplast. The light reactions are in the thylakoid membranes, shown here on the top in green, these sort of pancake-like structures. And the Calvin cycle, or the dark reactions, occur outside of those membranes, inside the chloroplast, in what's called the stroma. So you can see I've labeled by 1 the light reactions in the thylakoids, and 2 is the stroma, where the Calvin cycle is carried out. Notice that these thylakoids are in stacks, and these stacks are called grana. Each stack is a granum. So the light reactions go like this. We're going to have a very simple, a very broad view of what goes on instead of a detailed view. So here we go. First of all, light hits a chlorophyll molecule. This causes the electrons in the chlorophyll molecule to bounce to a higher energy level, and some of them are bounced off of the chlorophyll molecule. Chlorophyll doesn't like this in a sense, so it steals electrons from something, and it steals them from water. And this is how water gets oxidized. So this, aided by other processes, this causes the water molecule to fall apart, and that's called photolysis. Photo means light, and lysis means breaking apart. So photolysis is the breaking apart of water, giving off oxygen. So 1 water molecule produces 2 hydrogen ions plus 2 electrons plus 1 oxygen atom. The electrons and the protons, the hydrogen ions from water, are transferred to NADP+. This gets reduced. NADP+ plus 2 electrons plus a hydrogen ion result in NADPH. This NADPH has greater reducing power than water. It has more potential energy. Also, at the same time in this set of light reactions, ATP is produced. It's produced by adding a phosphate ion to ADP, and this is, of course, chemical energy. ATP is chemical energy, and this process is called photophosphorylation. That is to say, the addition of phosphate to ADP in the presence of light. So photophosphorylation. So we can summarize the light reactions in this way. Light energy is converted first to chemical energy, and that chemical energy is in the form of NADPH and ATP. Also, oxygen gas, O2, is released, and this, as we've said now, comes from water. Now, you can investigate photosynthesis at a very, very deep level. You can look at the details, we're only going to look at this in very broad strokes. So a very broad view of photosynthesis, and I'll try to emphasize what I would like you to know. So we're not going to look at the detailed steps of electron flow, linear electron flow or cyclic. We're not going to look at the electron transport chain. We're not going to talk about the two photosystems. We're not going to talk about how ATP is produced or how proton gradients are involved in that. We won't talk about proton pumps. We're just having a very broad view. So the light reactions, as I've just summarized them, and here they are again: water plus the energy of light plus NADP plus ADP plus phosphate gives oxygen, NADPH, and ATP. And if you see in this cartoon, in the upper part of this picture, there are the light reactions in the thylakoids, shown on the left inside this chloroplast, and then the Calvin cycle. And the Calvin cycle is on the right. And notice that the ATP and the NADPH produced in the light reactions are used in the Calvin cycle. And we'll turn to that in just a moment. But first, what's all this business about electrons and how are they moved and what role do they play? Well, they're important because chemical bonds are made from electrons. So transferring electrons enables compounds to be changed so that other atoms can be added to them. So how are these electrons moved in life? Well, there are two important kinds of compounds. One is NADP plus and NAD plus, and these are oxidizing agents. So they remove electrons from other molecules, including water, so they accept electrons from other molecules. NADP plus is probably new to you. This is the compound in photosynthesis that's an oxidizing agent. You've probably been introduced to NAD plus in respiration, which I'm sure you looked at in great detail. Okay, what about reducing agents? Those are NADPH in photosynthesis and NADH in respiration. These place electrons onto other molecules. NADPH in photosynthesis and NADH in respiration. So at the bottom, you can see the reaction, which can go in either direction. NADPH is produced from NADP plus 2 electrons plus hydrogen. A major feature of the light reactions is chlorophyll being hit by light. And this is what causes electrons in chlorophyll to bounce to a higher energy state, a higher level of potential energy. Chlorophyll is responsive to certain wavelengths. And here you can see what's called an absorption spectrum. And this absorption spectrum, or spectra for plural, is why plants are green. If you look at this diagram, the y-axis, the vertical axis, is the absorption, relative absorption by the pigments, and the x-axis is wavelength, so shorter wavelengths on the left and longer ones on the right. And this is visible light. So we go from about 400 to about 700 or so. And you can see chlorophyll A has a peak somewhere between 400 and 500, and also another peak up around the upper 600s. Chlorophyll B is slightly different from that. And then there's another pigment called a carotenoid. In fact, there are several kinds of carotenoids, and these have peaks down around the 500 or so range. So chlorophyll A absorbs mostly violet blue and red lights, and chlorophyll A reflects or transmits green light. This is why plants are green. And you can see on the right-hand side, there's a picture from your text that shows light going through, and green light is reflected, and green light goes through. And that's why leaves are green. That's why chlorophyll is green. They absorb other wavelengths other than green. So it's the reflected green that we see. So these accessory pigments, chlorophyll B and carotenoids, broaden the absorption spectrum. So here's the same picture on the top. It turns out that the absorption spectrum matches the action spectrum. The action spectrum tells us how much photosynthesis is occurring at each wavelength as measured by oxygen production. So you can see that it's highest down between 400 and 500 and also up in the upper 600s in the wavelengths in nanometers. So this matches the combined absorption spectrum of chlorophyll A along with chlorophyll B and the carotenoids. It's sort of interesting that photosynthesis occurs in the visible wavelengths. That is, it's not the energy of ultraviolet radiation, it's not the energy of infrared, it's not X-rays, it's not gamma rays. It's the visible light where the energy of photosynthesis is used. Notice also that shorter wavelengths have higher energy. So the blue light has higher energy than the red light. Okay, so the dark reactions. Here's Melvin Calvin. He discovered the dark reactions, the Calvin cycle, named after him now. He won the Nobel Prize for this work. The Calvin cycle makes sugar. It actually makes a precursor to sugar that is then turned into sugar, glucose. This set of reactions, the Calvin cycle, occurs in the stroma of the chloroplast, unlike the light reactions, which occur in the thylakoids. It uses NADPH and ATP from the light reactions. And the Calvin cycle supplies the light reactions in turn with NADP plus and ADP. So you can see that shown here in your cartoon in the lower left, where the Calvin cycle is supplying NADP plus and ADP to the light reactions. So how does this occur? What goes on? What are the steps? Here we go. Start at the top. Start around 12 o'clock in this diagram. In this cycle, the Calvin cycle, the fixation of carbon is accomplished by an enzyme. And this enzyme is called Rubisco. You can see the word written there. This is an extraordinarily important enzyme. It's the most abundant enzyme on the planet. Rubisco takes the inorganic carbon of carbon dioxide and adds it one at a time to a substrate called RUBP. And you can see that at around 10 o'clock on the diagram. RUBP is ribulose bisphosphate. Ribulose bisphosphate has 5 carbons. So each time a carbon from CO2 is added, it adds 1 carbon. So this produces a 6-carbon compound. If we do this 3 times, we have 3 of these 6-carbon compounds. They are very short-lived. These are quickly converted into a compound called 3-phosphoglycerate. And there it is. I've drawn a circle in blue around it. 3-phosphoglycerate is a 3-carbon compound. There are 6 of them in this cycle. This is a 3-carbon compound, so therefore this sort of photosynthesis is called C3 photosynthesis, because the first stable, fixed organic compound has 3 carbons. Then there are several steps involving the use of ATP, involving the use of NADPH. We're not going to go into any of those particulars, but finally we get to another 3-carbon compound called glyceraldehyde 3-phosphate, or G3P. Glyceraldehyde 3-phosphate is what leaves the Calvin cycle. So we have 6 of them in the cycle, 1 leaves, and you can see it there being shunted off down at the 6 o'clock position. And this goes on to be used in the production of glucose, which then can be used in the production of other sugars. So we have 6, we've lost 1. We have 5 remaining. So we have 5 G3Ps remaining in the cycle, and these go on and they have ATP added. They have various chemical reactions that then recreate RUBP, ribulose bisphosphate, up around 10 o'clock in the diagram. So this is kind of amazing. We started out with ribulose bisphosphate, and we end up with ribulose bisphosphate. So it's a true cycle. We have ribulose bisphosphate recycled, used, and recreated in the Calvin cycle. Additionally, we have carbohydrate output. We have carbohydrate made by the Calvin cycle that will be used in the production of other sugars. So, Rubisco, this important enzyme, takes carbon from carbon dioxide and adds it to the organic molecule RUBP. That's the Calvin cycle. So photosynthesis has the Calvin cycle and the light reactions. The light reactions are water and light producing NADPH, ATP, and oxygen. The Calvin cycle is carbon dioxide and NADPH and ATP producing a sugar, a carbohydrate, plus NADP plus ADP. So that's it. And here's sort of the level at which we want to understand the biochemical steps of photosynthesis. the light reactions and the dark reactions, the basic products, the basic inputs. That's really what we want to know. The very detailed steps, have a look at. Some of you might already know them quite well, but here we're just concerned with the broad strokes. Now, I mentioned that Rubisco is the most important protein in the world. I mentioned that it was the most common protein in the world. But Rubisco has a dual nature. Rubisco is like a Jekyll and Hyde protein. Rubisco is actually short for this very long name, ribulose 1,5-bisphosphate carboxylase oxygenase. That mouthful has been shortened to Rubisco. Why is it called oxygenase and carboxylase both? It's because of this dual nature. The carboxylase refers to its ability to put carbon from carbon dioxide onto an organic molecule. But it has this other property, and that is it can put oxygen onto an organic molecule as well. And which one it does depends on how much carbon dioxide and how much oxygen is in the area of Rubisco. Now imagine a plant that's photosynthesizing. It's using up water. The water is entering the roots, going up through the plant and out through the leaves. As it departs through the leaves, oxygen is produced. Oxygen is also leaving through the stomates in the leaves, and carbon dioxide is coming in. So while photosynthesis is occurring, water is being lost. So imagine now that it hasn't rained for a while, the soil is sort of dry, and that the plant is starting to dry out the soil around its roots. This means that it's going to start to lose some turgidity in its cells, and it's going to want to stop the process of water loss. How does it do that? So think of yourself now inside a stomate. Imagine that you're in a lecture hall, and the top, the ceiling, is the upper part of the leaf, or the upper part of the stomate. It's filled with wet cells. The sides, the walls are filled with wet cells. The front and back, wet cells. The floor, wet cells, but two of those cells are the guard cells. And those guard cells are the sausage-shaped cells that open and close. So if the plant senses dryness coming on, it can close its stomates, and that prevents water loss. The prevention of water loss, however, means that the stomate is closed, so if photosynthesis is still occurring, if it persists, then we have the buildup of oxygen inside the stomate and the reduction of CO2 concentration. This means that the relative quantities of CO2 and oxygen are changing. Oxygen is going up, CO2 is going down. Once this happens, Rubisco switches from taking carbon dioxide and adding it to RUBP to taking oxygen and adding it. This is a problem. This causes the plant to waste energy. This causes the plant also to make no ATP. So we have a problem. What does the plant do? What about plants that typically live in these dry conditions? This addition of oxygen by Rubisco is called photorespiration. It attaches oxygen to RUBP. This metabolic pathway consumes oxygen. It releases CO2, so it's like respiration in that regard. However, it makes no ATP, so it's not true respiration. It wastes energy, and it decreases the photosynthetic output of the plant. Organisms have invented a way around this. Some organisms have an enzyme called PEP carboxylase. This PEP carboxylase is also referred to as PEP-C, and it is prominent in two kinds of photosynthesis. The first of these is called C4. C4 photosynthesis occurs in about 3% of land plant species, and it has a particular anatomy associated with it. So you can see here a vein that has the vascular tissue in it. Outside the vascular tissue is something called a bundle sheath cell. And then outside of that is a mesophyll cell, just a general sort of parenchymal cell that has chloroplasts in it. Now, if you look at the right, you can see a magnification of these two cell types. So mesophyll on the top, bundle sheath on the bottom. And this sort of anatomy is called Cran's anatomy. It means wreath. So it's like a wreath if you look at this in cross-section. And if you look at the mesophyll cell, you can see that carbon dioxide is coming in through a stomate, and instead of meeting rubisco, it meets PEP carboxylase. So PEP carboxylase then makes carbon dioxide, adds it to PEP, and makes oxaloacetate, OAA. This is a 4-carbon stable compound, oxaloacetate. So this is where this photosynthetic system, this photosynthetic process gets its name, C4 photosynthesis, because of the C4, the 4-carbon oxaloacetate being the first stable carbon compound made from carbon dioxide. The oxaloacetate is then turned into malic acid or malate, and the malic acid is shunted into one of these bundle sheath cells. The bundle sheath cells are away from the air, away from the atmosphere, protected from the atmosphere. So once the malic acid goes down into the bundle sheath cell, that malic acid is converted into CO2 and other materials, in this case pyruvate, and this CO2 then enters the Calvin cycle in the normal way. So RUBP, with the help of rubisco, has carbon dioxide added to it, and the Calvin cycle proceeds. This sugar is then produced and it goes into the vascular tissue where it can be moved to other parts of the plant. So the main point here is that C4 plants have a spatial separation of processes. Carbon dioxide enters the plant in the mesophyll cell and is acted upon by PEP carboxylase and not by rubisco. So even if the stomate is closed, the plant does not add oxygen onto RUBP. So PEP carboxylase has the wonderful feature of not adding oxygen. It doesn't have this dual nature like rubisco has. So it won't add oxygen onto RUBP even if the oxygen concentration gets high and the CO2 concentration gets low. So that's why this is an adaptation for plants in dry regions. It occurs particularly in tropical grasses like corn and sugarcane. So the Calvin cycle then occurs away from the atmosphere down in the bundle sheath cell. Here's another picture of the same process where the book has written out in words the steps. So have a look at this if you want to. It's basically what we've just said. So here's a summary of what I've just said. In the mesophyll cells, carbon dioxide enters and water exits. There's no rubisco. And in the bundle sheath cells, there's rubisco, and this is kept away from potentially low carbon dioxide, which is an adaptation for life in dry conditions, where stomates are often closed. Now there is a second adaptation for dry conditions, and this is called crassulacean acid metabolism, or CAM, C-A-M. And CAM plants are familiar to all of you. They include the cacti, they include agave, they include the jade plant. CAM plants make up about 10% of flowering plant species. And it's shown on the right here. Here's a plant called hens and chicks. And in contrast to C4 plants, CAM plants have a temporal separation of steps. So at night, these organic acids are produced, and in the daytime, the organic acids are turned into CO2 and the Calvin cycle proceeds. So these plants open their stomates at night, they take in CO2, PEP carboxylase is added, and organic acids are produced. In the daytime, these organic acids are turned into CO2. So if you walk through a desert and you're tasting a succulent plant, don't try a cacti because that's just a bad idea. If you're tasting a succulent plant and you start out in the morning and you notice how acidic it is, you will notice that it's quite acidic because through the night, the acids have built up from the fixation of carbon dioxide into these acids. It happens through the night while the stomates are open. Then when the sun comes up and the day starts to get blazing hot, the plant has closed its stomates and it's turning these acids into carbon dioxide. So during the day, you will notice, if you keep tasting these plants, you will notice that the acidity drops. So you know CAM plants. Here's a cactus. Cacti have gorgeous flowers, many, many petals. Here's a plant. This is called blue agave. Blue agave is famous because it's the source of tequila. Here's how you make tequila. You get some agave leaves, these great big long succulent structures, and you cut them off and you dig a pit in the ground and you throw them in the pit, these leaves in the pit, and you build a fire in there and you cook them on down and you just let them roast, and this brings out the sugars in the leaves. And these sugars, it's like cooking onions slowly. It brings out the sugars. And it brings out the sugars, and then the yeast like these sugars. So there are natural yeasts hanging around, and these yeasts will turn the sugars into alcohol. And so do this in a big pot. Just do this for a while, and you'll build up the alcohol content. And then you take this juice and you distill it. And the more you distill it, the finer the tequila is, and the more you can charge for it. So that's how you make tequila. This is pineapple. Pineapple is another famous cam plant, another succulent. So that's photosynthesis. This is an overview of the steps that I'd like you to know, an overview of the light reactions and the Calvin cycle. There are 2 adaptations for dry regions. These are C4 plants, which include many of the tropical grasses, and cam plants, which include basically all the plants you think of as succulents. Next time, we're going to turn to the control systems in plants. See you then.